Heard from these symposia, I think it's always proper to talk about Irvin Mueller's work. So if I can have my first slide. I guess you can't see that. Can you focus that a little bit? Can you all see that? Okay, this is a slide I took from a review article of Irvin Mueller, and where he looks at the imaging of molecules. And this was one thing which excited my interest, because I have become interested in biology, and the question is, what could I do in biology? So I said, well, one thing I possibly could do is try to compete with Mueller and make a microscope you can see molecules. And you see here, Mueller has an electron emission. He has all different kinds of molecules. I think Irvin got carried a little bit away with that mystical thing over there. But nevertheless, it is possible to see molecules by using a field emission microscope. The question is, why would you do such kind of research? And on the next slide, shows you what I call the molecules of life. The question was asked, what use do these things have to biologists? Well, biology, of course, is a very complicated subject. And since hopefully there are no biologists here, I have taken the liberty to reduce the complexity. So life really essentially consists of water plus 30 precursor molecules. Those are the most important molecules in biology. They have some ions here and there and stuff like that, if you will bear with me. So these are the 30 precursor molecules. And if you let me start from the bottom, what you have is fatty acid, and you have trialcohol like glycerol, and an amine. These are the building blocks for the membranes which surround the cells. And I'm really not going to say much about those. Then we have the 2D sugars. But you know, not all molecules, but many molecules are not symmetrical. They can appear in a left-handed and right-handed configuration. And in biology, in life, the sugars always is the D sugars. So if you're going to look for life on Mars, for example, what you would do, you will see whether the substance you looked for were polarized. If it's polarized to light, then you know, you're pretty sure that there's life there. If you shine light, for example, for corn syrup, you'll find out it's polarized when it comes out on the other end. Because they're always one molecule, and not the optical isomer is not made in light. Now, what sugar makes, they make cellulose, starch, and glycogen. Glycogen is a way for animals to store energy, like starch is the way that plant stores energy. And basically, these aren't really interesting in biology. People think they know the structures of these things. Then, of course, we get to the two big items. One is the RNA or the DNA, and the other one is the proteins. And those is where that is chips where the action is. So you have protein. The protein consists of L amino acids. Again, the two configuration, L and D. Only L amino acid consists in nature. And the RNA, or the DNA, has 5 different bases. And they build up, they are all polymer molecules build up on these bases. Now, on the next slide, I show the principle of DNA. And the principle is as follows, is that you have 4 different molecules. And the structure of these molecules are well known. You can get that from X-rays. No point looking at those. You know what these structures are. They aren't squares and circulars, of course, but I mean the structures are well known. Now, when you make a DNA molecule, what you do, in a single-stranded molecule, these bases are stuck together in a long strand. And the information is in the order of these molecules along the strand. That's where the information is. And the DNA molecule will eventually make the protein molecule that makes the life processes go on. But what you inherit is a molecule like that, and that is which tells you how you would look like and how your children would look like and so on. Now, the DNA molecule is very interesting because it's the only molecule which will replicate itself. No other molecule will replicate itself, and there's a big race to try to find some chemical or some model system where you can make it replicate itself. What the DNA molecule does, it makes a complementary strand, such that the square always goes with a circle and the triangle always goes with a cross. So this molecule will make a complementary strand which again then, of course, will replicate the original molecule. And the problem with this replication is that when the replication goes on in life, you need enzymes. And an enzyme is a protein molecule. And the protein molecule has to be made from the DNA. And therefore, you see, of a circular reasoning, what came first? The DNA molecule or the enzyme? Well, nobody knows. I mean, like somebody says that a chicken is an egg's way of making another egg, you see, right? There's a question of chicken and egg, and here's the question of the DNA and the enzyme. You know, which is the primary and which is the secondary. And original life is very important in a very important problem in biology. A lot of people speculate on that. So anyway, this is the principle of the DNA, is that the order of the bases is what's important. Now you can see, of course, DNA molecules in the electron microscope. And a very famous picture is shown in the next slide. That was not. Can you have the next one, Jerry? Either I got mixed up or you did. That's the one I was looking for. Here is a small virus called the bacteriophage, is attack bacterius, and is just a protein envelope, and that has been blown open, and the DNA molecule, which is this long strand which you see here, all at one time fitted inside that little virus. You see, DNA is the longest molecule known to man, and it's an amazing thing that you can have this virus contain all that information. Now, if you look at that picture, of course, you say, well, there's no way that molecule can get back into that little virus, and the reason for that, of course, is the molecule has been stained, as the previous speaker talked about. So since it's stained, it's much thicker on the picture than it really would be if you hadn't stained it. So it's probably enclosed in glass or whatever they use as a stain. Now, you see a picture like that. Actually, another thing, if you look at this molecule, there's sort of a cocktail party conversation. Each of your cells has a molecule or several molecules of DNA in it, and if you stretch them end to end, they will reach across the solar system easily. So you see, you have an enormous amount of DNA in your body. And now, of course, you would like to know some information about this molecule, and of course, you can't really see anything in these electron transmission micrographs. But you can get the structure from X-rays, and on the next slide, we get the one that we had, which you took out. This is of course what Crick and Watson did. They figured out from the X-ray what the structure of the DNA molecule was, and it is a double helix. And they also figured out that the basis was combined the way I showed on the first slide. And now, the previous speaker asked the question, is it any use to see these kinds of structures in biology? And really, it would be a great feat if you could see a picture of a double helix directly, but it wouldn't be any use to biologists. Because we know, can I still have that picture, Darren? You keep disappearing here. I do like to keep that a little bit. We know from X-rays that this is a double helix. And therefore, if you could take a picture, by an electron microscope or some other means to see the double helix, that would be nice, but it wouldn't be much of use because we already know what the structure is of this molecule. What would be of tremendous use is to find out these are the bases here, find out which base is where along such a strain. That is where the information is, and that is what biologists like to do. And they can do it now chemically. And on the next slide is shown a virus which has the sexy name IFES1978. And the virus looked like that, looked like with some object from outer space. And this virus has inside it a DNA molecule. And last year in Nature, the structure of that DNA molecule was published. And this is the publication. You probably can't see it from there, but these are the letters from the code. And it turns out it has 5,375 bases in that particular molecule. It's just an absolutely amazing feat, and that they can do chemically. But to do it chemically, you need a large amount of stuff, and that's why electron microscopy or feline microscopy or something like that could have an advantage because they could do it with much fewer molecules. Could you just leap for the other two? See, there is more information, and there's even one more. And it's a really absolutely amazing feat was done by Sanger in England. And if you know the complete information contained in that virus. And as I told you, it sort of looked like an object from outer space. And when people send objects to outer space, what they tend to do is put a picture of a man and a lady and maybe Nixon's voice on it or something like that. And I really think what they should do, they should send the code, which we know for that particular virus. And now if the aliens, if the code is really universal, they can then reassemble, they can remake the virus out there. Now then, of course, they will start wondering how this virus can be so goddamn smart, right? They would think it was a picture of our own image, which of course it's not. But sometime in the future, I think possibly that people will send the DNA code for a human being. That's not an unthinkable thing. It's as far in the future, somebody has to find some clever way of sequencing the DNA, but it certainly is possible. Now, on the next slide, I'm going to talk a little bit about protein. Now, DNA is what makes protein molecules. And protein molecules is what makes the chemistry go on in your body. And the protein, of course, is a common name for a whole class of macromolecules. For example, all the known enzymes are protein molecules. There may be some which are not, but all the known enzymes that keep you alive are protein molecules. Many hormones are protein molecules. And protein, of course, is a large molecule. The molecular weight is typically 10 to the 4 to 10 to the 6. And as I said, it's a polymer molecule built up of monomers called amino acids, and there are approximately 20 of those which exist in life. And on the next slide, shows you such a, what they call a primary structure. That's finding out which amino acid is where along a peptide chain. So that's very similar to the DNA. DNA was a long chain molecule. Protein is really a long chain molecule. In a typical peptide chain, you may have about 100 amino acids in it. And the basis for the primary structures, you have to find out which molecule is where along such a chain. That's what people want to know. And you can do that chemically, and it was first done for insulin by no other than Sanger, who did the DNA molecule I just showed you. He did that back in 1953. Insulin, of course, is a small molecule. It has two chains, one with 21, one with 30 amino acids in it, and he established uniquely what the primary structure was of insulin. And that was the first time people knew that insulin was a specific molecule. Of course, they suspected that before, but nobody knew that my insulin looked like your insulin, looked like somebody else's insulin. Insulin is always the same. You take insulin from an animal, it's a little bit different from people, but not much. And people with diabetes very often take insulin from animals to make up for the deficiency of not having insulin. Now, on the next slide, is shown that when the protein is in an active state in the body, when it acts as an enzyme, the protein is curved up on itself in some sort of way, like a bowl of spaghetti. And that some sort of way is called the active state. Sometimes it's held together by disulfide bridges. Sometimes it's just hold there because it's in the lowest energy state. And you know it's in the lowest energy state because you can take that molecule and very carefully denature it. And you can sort of stretch it out, and you can do that by heating or by acid or some other way. Then you can slowly cool it down, and it will reform and take its original shape. Therefore, the primary structure of that molecule determines the three-dimensional shape. And in principle, you can calculate, if you know the primary structure, the three-dimensional structure. But nobody's been able to do that. And you see, if you sit down and calculate, you go through all configuration, it turns out that even the biggest computer will take hundreds of millions of years. So you have to find some shortcuts. You cannot calculate all configuration possible. You have to find some shortcuts. A lot of people are working on that problem, but it's a very difficult problem. And the primary structure is very important. For example, there's the hemoglobin is known that it consists of 2 alpha and 2 beta chain. each having approximately 150 amino acids in them. And in one of those chains there is an error, and these people, where the error occur, they have sickle cell anemia. And sickle cell anemia is a deadly disease. It's a molecular disease. Linus Pauling coined that term, and it's a molecular disease in a sense that you are inherited disease. There's nothing you can do. There's something wrong with your DNA. But we're going to fix up the DNA first. It's some genetic engineering that's going to get rid of the disease. And I'm sure that a lot of molecular diseases exist, but we just don't know about them. Because to look at these primary sequences, is a long and tedious problem. And if you had some simpler way of doing that, it would be, that would be a great advance in protein chemistry. You could do it fast and cheaply. Now, on the next slide, it shows a picture of a protein molecule. This is myoglobin, which is similar to hemoglobin. Myoglobin is a protein molecule which stores oxygen in muscles and things like that. And here you see you have the whole, this is the front view, this is the side view, and you have the whole three-dimensional structure of this molecule. And the chain goes, you can see this corkscrew thing going along here. These corkscrew things are the chain we curl up in what is called the alpha helix. Pauling was the first person to point that particular structure out. So this molecule consists of an alpha helix there, one there, and so on. And the alpha helices are referred to as secondary structure. The full three-dimensional picture is for the tertiary structure. And you see this particular molecule, its structure is made like a box. Because really what it contains is a heme group, and this heme group stores the oxygen molecule. So really the function is very much fitted to its shape. And it is to get a picture like that, I don't know, let me see if I can guess, it probably takes you about $10 million. Because the previous speaker said you had to do the x-rays, and it takes an awful lot of computer time, awful lot of hard work, and you have to have a lot of different molecules available. And most molecules, of course, you can't get them. That's why myoglobin and hemoglobin are so easily to work with, because they are so plentiful. And that's why these molecules are first to get the three-dimensional structure first. But if you can get the general shape of such a molecule, that would be a valuable information. On the next slide, shows the side. This is a hemoglobin molecule, which you have in your blood, and that really is very closely related to the previous one I showed. There are really four of them stuck together. But let's show this to show you the size. This is 64 angstrom in size, and compares to the heme group, for example, which is 12 angstrom, the benzene ring, which is 6 angstrom, or water, which is 2-3 angstrom, or 4 angstrom it says up there. So you see the molecule, protein molecule, is very large compared to, the small molecules we are as physicists, we are used to work with. And as I said again, if you, by electron microscopy or some other way, D-line microscopy, can get the general shape of such molecule, that would be valuable. What they do at the present time, they interpret light scattering, diffusion, and centrifugation, and things like that, to try to get the general shape of the molecules. They cannot make X-ray pictures out of them. Now, I have been, I'm going to tell you a little bit, I told you I've got interested in repeating some of Mueller's work and look at molecules in a feline microscope, and so I got interested in how protein interacted with a metal surface. Now the next slide is showing the particular surface I use, and what you see here is a little dirty, but what this is an indium slide. It's simply a piece of glass on which I've evaporated indium. And indium goes down in little particles. And a little particle scatters light, and that's why this looks brown compared to the rest of it here. This is not a picture, by the way, it's a real indium slide in there. If you look at that indium slide with an electron transmission microscope, you find indeed that the indium consists in little globs. And these globs are approximately 5,000 angstrom in diameter. And I use this slide to tell people I have become a real biologist, because you see there's no scale on that slide. And if you look in the biologist's book, one thing that irritates me is that they never put scale on their pictures of cells and things. Scale in biology really isn't that important to them, not to a physicist. They're more important in function and form than scale, because, you know, old cells aren't the same size and things like that. Now, I thought what I'm going to try to do is put on a little demonstration for you here. That's also the slide, Jerry, for the time being. If I can get this one to work. Did anybody know the secret formula? See, what I have here is I put down on the slide here. As you can see, those are the real thing. And they show any kind of stuff you put in the slide shows up very well. If I press my finger on it, you see my fingerprint. And if you wait for a while, the fingerprint will sort of diffuse out because the oil diffuses very fast. What I'm going to try to do now, if my hands are steady enough, is put some protein on the slide. You're going to see if you can see that. It's almost like, if I drank it now, right? It was good stuff. And this thing here is a bottle of human serum. That is blood, but they have taken out the cells and some clotting factors. So it contains all the protein in human serum, and there's approximately 100 milligrams per ml of protein in it. And if you take a little bit, what you need is something like 1 milligram per ml is all you need. So, but it doesn't make much difference. And amazingly enough, if you look at human serum, and I came as a physicist, I thought that everything was known about blood. But people don't know what's in there. They know a few molecules, but there may be 10,000 or to 100,000 different molecules. People have no idea what they are. And you know, I mean, they know about the most common one, but not a lot of them they don't know about at all. What I'm going to do now, I'm going to take a slide like this, which there's nothing on it. I feel like, nothing on my sleeves, right? I'm going to dip it into the human serum, halfway down, and take it up, and hopefully try to use Mrs. Harnet's flour thing to wash it off here without making everything too wet. I stole a lot of napkins from the cafeteria. And now my claim is that protein will absorb on that slide, and it will absorb only in a single layer. Doesn't matter, make a difference, so long you have the slide in there, you only will get the single layer of protein. You get a monolayer. And there it is. You see where that slide is dipped in. You see it dark, it's a little wet, but you see that's where the human serum has absorbed. So what you're looking at there is about 20 angstrom worth of protein. That's all. So you see, your eye is pretty good, you know, when you knock yourself out to look at, you know, thickness, 20 angstrom is a lot. Now, if I could go back to the slides. What I did, actually, John Penn has got me onto this because he had trouble with water. So what I did here, I left a little bit of the General Electric tap water on the slide and let it dry out. And that's what we drink every day in Schenectady. And if you take triply distilled water, coarse distilled water, you see you still have some stuff left in the slide. Of course, what you're looking at here is mostly salt. You know, if you washed it off, it'll just rinse off. And on the next slide, I made this layer. This is bovine serum albumin. It's a pure protein. I wasn't sure if I was going to do this demonstration or not, so I'm going to show you that the protein indeed shows in the slide. And what you're looking at again is like 20 angstrom worth of protein here versus nothing there. Now the reason you can see it is that the protein is on small metal particles. And the next slide, shows is a guy named Schraffman who back in 52, I guess, did this calculation experiment with microwaves. He used a large sphere and he looked at the scattering versus the diameter, and you get some sort of curve like that. And then he calculated the scattering versus the wavelength, and indeed you get the same for calculation. But then he put thin layers of dielectric on his spheres, and he found out the scattering changed quite a bit. And again, he could calculate it because he dealt with a single sphere, and calculation experiment fits very well. Now what I'm doing is exactly the same thing, except I use light, and I use much, much smaller spheres. And my dielectric is the protein. And the protein shows up in these spheres very well. It will not show up if you take a piece of glass or a piece of metal or something like that, because the layer is much too thick. You need these little spheres to become visible. Now, I'd like to tell you a little bit about immunology, because that's what I'm working with, and I'm going to show you how I apply that to John and I have done that with the field emission tips. And so let me give you a few terms about immunology. And the next slide shows some of the definitions. Immunology is simply an adaptive defense mechanism, right? And an antigen is any foreign material, and an antibody is a specific protein molecule made by the body to combat the foreign material. And the best way is really to talk in examples, because I don't learn from reading general things. I learn best if I talk about examples. And I gave this a talk similar like this in India a few years ago, and this is a very interesting part, because now I can choose to talk about syphilis or chickenpox. And last time I was out here, I chose chickenpox. But now I know these people a little bit better, so maybe I should talk about syphilis, I don't know. It's probably safer to stick to chickenpox. The point is that chickenpox is a foreign material. It's an antigen. You will get a chickenpox in your system. The chickenpox, the virus, will multiply and destroy your cells, and you will get ill. Now, while this goes on, your body itself would make a protein molecule called an antibody. And the antibody is made to attach itself specifically to the chickenpox virus. That's the only thing. It will attach to that specifically. It's made for that purpose, somehow by the body. And when the antibody attaches itself to the chickenpox virus, your white blood cell would then eat up the whole system and get rid of the chickenpox virus. It's like you label them with a label. And that's what your antibody, that's one main function of the antibodies. And so then you get well, and next time you get chickenpox virus, you don't even get sick, because you have the antibodies and the memories of making them, so you don't get ill the second time. And that's how all vaccine works, for example. Now, people vaccinate, you take an altered virus, and you vaccinate, you have the antibodies, and now you have the antibodies to take against polio. When you get the real polio virus, you won't get sick because you have the antibodies there. So these antibodies are very important molecules. And nobody as yet has an X-ray pictures of these antibodies. They are getting finer and finer resolutions as I go through meetings, but you don't know what they actually really look like in full detail yet. Now, on the next slide, it shows you sort of a physicist simplified view of this thing. Here you have an antigen, which is any kind of foreign molecule. And here you have and they have certain sites on it called antigenic sites. And here you have the antibody. The antibody I'm talking about always look like a lobster. They know the general shape, with two claws. But their final shape, this antibody attaches to this antigenic site, is slightly different from that antibody attached to that antigenic site. And the body somehow knows how to make the right antibody when you get challenged by an antigen. And if you have the antibody for polio, you still get chickenpox. If you have a chickenpox, you can't get any of those diseases, and so on. And now you see, it's a very nice thing, because if you have a protein molecule, whether you like it or not, you have your whole medical history in your blood. So if I could hold on to the protein molecule, stick it into your blood, and if anything attached to that molecule, it has to be the right antibody. If I take the antibody and wave it around your bloodstream, and anything attached to it, it has to be that molecule, because the reaction is very, very specific. Now, that's an amazing thing, but that's the way it is. And that's the way the medical profession looks for illnesses and things when they look at your blood. You get married in New York State, you got to be tested for syphilis, for example. If you have syphilis, I don't know what they do to you. You probably still get married, but you haven't been tested anyway. Your blood transfusion has to be tested for hepatitis, for example. This sort of things. Now, let me show you how I do this with my system. I think I have a view graph here. And this is in principle how you can build up a double layer of protein molecule. You take stick a slide or any piece of metal, matter of fact anything, into a protein solution, and you find out that you will get the monolayer of protein out of it. And this happened to be bovine serum albumin, which is a major component of the protein in the blood of a cow. And you get a single layer on that surface. Now if you take this, if you leave the single layer of BSA there and stick it into serum, and serum has, as I said, hundreds, maybe 10, at least 10,000, maybe 100,000 different protein molecules. None of those will go down and settle on the original layer. They will not. But if you do the experiment again, but now you stick it into a serum where you have specific antibodies to BSA molecule, the antibodies will go down and settle down, and you get a double layer of protein. And the way they do that, they take bovine serum albumin, which is from a cow, they inject in a rabbit, the rabbit makes antibodies to it, and now these antibodies will then later on be able to go on the surface. You get blood from that particular rabbit. And let me show you now on the next slide. Just skip that one, the next slide. What I'm going to show you is a demonstration of how these slides work, and it's sort of what I call fun and game, because a General Electric company, whether you have a Nobel Prize or not, you got to talk to your manager every year and tell them what you're doing. And that's when creativity is high around the lab. And what I did to explain this particular system, I wrote, I hope you can see it, it says BSA with a molecule BSA on one of these slides. And this is a real slide you see now, it's not a picture. And so you see here something like 15 angstrom letters on the background. Now you take that slide and dip it into serum, the next slide, where the rabbit, this particular rabbit, do not have antibodies towards that molecule. And therefore the protein from the serum will deposit themselves around the letters BSA. And the letters is still there, but you can't see it because you have the similar thickness of layers surrounding the letters. But now if you take a slide like this and put it into a serum where they have antibodies to BSA, the antibodies go on, and here you get the double layer. So here you have a thicker layer, and you can see that compared to, this is a double layer, that's a single layer. And you see this is a relatively very easy way, or simple way, which you can do immunology in the hospital. And so we're trying to do that, and going after some of these kinds of markets, but a very difficult thing to do because, you know, there are several other simple immunology tests. But the medical profession like this very well because they say they have to contend with nurses who aren't very smart, so what they envision, you can write down the slide what kind of fails you, you dip it in the blood, and it just shows up when you peek fruit and you know sickness. And I told them they could put the bill on the slide at the same time, and they would like that's one thing they understand very well. Now, so when I was doing this kind of work, and then I got together with John Panitz, because my original interest was to look at these tips to see that you could see these protein molecules. And I wish I could say to you that we see 20 pictures, but they really don't have much information in them at the present time. And we have gone through a whole series of errors that happen very often in your research. You have to try to do the right thing and the wrong thing and whatnot. And I'm going to show you something you have to avoid when you're going to try to stick something in a tip, and that is you have to avoid phosphate. Most protein chemists use phosphate buffer to keep the pH around 7. And it turns out that if you use phosphate buffer, you simply take the protein off the surface. You see here I put the protein on, and I put phosphate buffer on, and actually remove the protein from the slide. And I had this brilliant idea of making a General Electric dishwashing detergent, but I don't know if you know or not, but phosphate is illegal in dishwashing detergent now because it's a pollutant. And nobody really knows why phosphate take protein out of the plate so well, but it does. It's a well-known fact in the dishwashing industry, but not so well known if you're a protein chemist, because they basically don't deal with surfaces and protein. So anyway, so we had to avoid that. We had to avoid several things, but let me show you a few needles where we have a protein layer on it. This is a nominal 1,000 angstrom, not exactly, and this is one of John's needles. I don't know whether he wanted to recognize it or not. And you can barely see, at least here where I stand, that you have a protein layer on this needle, but just barely. This is bovine serum albumin. Now, on the next slide, I show a layer you can see much easier, and that's ferritin, which Isaacson also talked about. Here you see the needle, and here you see the ferritin molecule in a single layer on the side of the needle. And you see, he said they contain 5,000 atoms of iron, and presumably that is what shows up so well in the microscope. So here you can very easily see that you have a single layer. I don't know what that is, but basically it's a good, nice single layer. So it's possible to get it on the tip. On the next slide is, I wish I could have said it's a typical tip, but it's an atypical tip. This is what we would like to see. What we have done here, it puts BSA on the tip first, which you barely can see, and then you expose it to an antibody. So here you have a double layer of protein on the tip, and you see a very nice, even double layer all the way around. And miracle of miracle, if you measure the thickness, it agrees well with what you can measure by other means. And so you actually can see this particular double layer. And this is really what Jan had been striving for, getting protein on the tip reproducibly. But I said, I think this is my first attempt, and after that, they always look like in the next slide. You see, this is really what you tend to get. You get these enormous thicknesses, which you don't know really where they come from. And you get uneven buildup and everything else. And it's a very, very tricky problem to do reproducibly. And since it worked so well with my slide, I was sort of misled. I thought that protein will stick well with anything, and I have to now change my rules. They stick well with everything which is flat. These needles really are too thin, and I'll show you what I think happens. Enough of the slide for a second here. If you have a tip which is small in diameter, like you feel-eye microscopies have, and if you take it up from the water, you would think you get a water layer. Say it was infinitely vegetable, you think you get a layer like that, but you really don't. This is an unstable configuration. What you do get is a layer like that. So the tip really comes up almost dry, and the water sort of snaps back. And the surface tension you have will remove the proteins from the tip. Sometimes remove them so you get a big deposit here, sometimes get the other way. It's easier to see. I tried it out experimentally, and but using a flat surface, let me show you really what the physical principle is. If you have a flat surface of glass, and here you have the double layer of protein molecules, if I stick this slowly into a solution of water, what will happen is that the water will creep up like that, and you hear the protein likes to be at the air-water interface. So what you do get, you get a mechanical force. It simply tears them off the surface if you do it slowly. If you do it fast, it doesn't happen. If you do it slowly, it happens. And apparently with needle, it's always happened. The last slide shows that that is indeed what happened when you do that. Here is a protein molecule, and here is a full double layer. This side has been dipped into water from here and up, and you can see that it looks much lighter here than it does that there, because we have removed some of the protein molecules. And here we have deposited a amorphous kind of layer. You can see the very sharp thing at the water air interface. And somehow that must happen when we deal with these small tips. That's all for the slides. If I can get a light back on. I'd like to conclude then to say that I sort of came into biology because I became interested in whether protein, I was very interested to see if I could make a microscope which I could see protein molecules, and to do that I had to find out how they interacted with metal surfaces. And now really I made it my major research project is the interaction of biological molecules with surfaces. But I was fortunate enough to team up with Jan, so he's taking care of the microscope aspects. I didn't have to worry about that. So now we are still on the trail of trying to make some deposit protein on the tip reproducibly. And Jan is going to talk a little bit more in detail about that later in the session. If there's any questions, I'll be happy to try to answer.
comment you were mentioning about the water experiment. It turns out there are very few techniques that will detect impurity levels in water as accurately as these slides. Good gas chromatographs, for example, show nothing when you get concentration levels as low as in that distilled water. So these slides are really a very impressive technique to show layer thickness.
What was the metal used?
Apropos of the difficult problem of getting proteins to stick to field iron points, I really don't know how many different materials you've tried. Have you, for example, tried indium as a tip material?
No.
That was much more close to your actual.
Yeah, that's true. Now the metal on the slide on indium, the reason that indium is it's very easy to make little particles of indium. You simply just evaporate it and it forms into little particles by itself. We have not used indium. Jan has restricted at the present time to tungsten.
We use gold, tungsten, molybdenum, both annealed and non-annealed. Gold because it doesn't have an oxide layer or a small oxide layer. The annealed versus non-annealed have to do with the smoothness of the surface.
If you do different kind of metals, it turns out that, as a rule of thumb, protein sticks to everything. And all the metals you use, protein will stick to, not necessarily equally well.
Why do you want to have a double layer on your tip?
Well, we don't really want to have a double layer in the tip, but the single layer you can see so thoroughly. And I personally am interested in double layers. And if I can see what the sizes are, because I've given the talk, I talk to biologists, sometimes they don't believe I have a double layer. See it, because that's one of my interests. And we know if we can put a double layer on the tip reproducibly, then we have a problem of lift. We can see that even thickness. We can do that reproducibly. If we can't do that reproducibly, if the single layer isn't reproducible either. Yes?
You had some field and temperature. What's the temperature you use?
The temperature?
Yeah.
When
you're putting the protein on, what's the temperature? How do you do that?
at room temperature. These are done at room temperature.
Why do you try to heat the tip instead of trying to have the room temperature?
I'm sorry, I can't hear. I hear very
If you
badly.
try to heat the tip higher than the room temperature, what would happen? You get a uniform layer of protein on it?
They got higher than room temperature? Well, basically with protein, you're limited to something like 40 degrees.
Yeah, so you can go up to 40
You can go up to 40 degrees C. And if you go higher than that, protein will denature very badly. So you could go higher up, but you haven't really done that. You basically done everything at 25 degrees. Yes.
You put a little bit of water on the side of the needle and let it sort of diffuse up around there, that you never have this withdrawal effect. Do you think that might work?
It might. We did a little bit putting the needle in the microscope. If you put water on it, it doesn't do anything. Now, if you breathe iodine on it and watch it in the microscope, when the ice melts, as soon as the ice melts, it snaps back, very fast forward. So it may very well be possible to do something like that. Yes.
Is there anything you can put into the water to break the surface of like nonstick or something?
Yeah, that's a good suggestion. And what we have been doing is putting alcohol into water. That's a bad thing to do. It turned out the surface tension of alcohol is about 1/3 of water, and that helps some. And I should have said that these pictures here, as I showed now, they were taken by, except for the one, which was the best, they were taken by putting the protein on, then washing the protein out with clean water, and then putting alcohol in, and then taking the tip out. And that seems to help, but these are one of these things that since you're relying on the electron microscopist, essentially the enemy, right? It takes time to do, so we haven't looked at more than maybe 30 tips or something like that order. So you're not sure. I mean, it looks like it helps, but I'm not certain it helps. Yes.
Is it not possible that to evaporate on the tip something which reduces the surface tension of the tungsten?
Yeah, that may be possible, but then you probably have a difficulty with putting a field line microscope to try to to look at it. But it may be possible to put some other materials on it. That's true. But we really would like to have it on a clean metal surface.
If this is really a desperate problem, you could always go to a primitive one. Are you familiar with the method, the electron microscopies used.
No.
You go from, well, I can talk to you later.
Sure,
but
sure.
It's going around the critical point.
I see, right.
So you never go through
I heard about
a
that.
water interface.
Yeah, you effectively pressurize
Oh, I
the
see,
vessel
right,
so
right.
that you now gradually reduce the water volume and you have a gas like CO2.
Oh, I see what you're saying, yeah, I never thought about that,
It's-
maybe John had.
It's been developed, it's quite a practical method.
Right.
And,
Okay, well, see, we really can't really put the molecules on the tip reproducibly, and as I'm sure you all know, in feline microscopy, you take a long time to put a needle in your microscope and things like that, you can literally weigh weeks and months, and as you know, that the first problem with the molecules are there right at the tip.
Okay, no more questions. Thank you once again.